types of genetic mutations that can happen. In the first model, we have 4 different strains of DNA that have been transcribed and translated into amino acids. The first question asks you to look for nucleotides. A nucleotide is made up of 1 sugar, 1 phosphate, and 1 nitrogenous base. Normally, though, you will only see the letters in a DNA sequence of these types of questions, so you would determine the number of nucleotides by counting the number of nitrogenous bases, which in this case is 21. Codons is a word for 3. When we talk about codons, we're talking about the mRNA sequence, and we're talking about 3 letters as a group. So you need to count the groups, which is 7. Sorry, 6. Doesn't include the stop. When it is asking you for the number of amino acids, it's asking you to count here how many different things make up this polypeptide chain. It is also 6. In the next question, it asks you for the sequence of amino acids. It literally wants you to write out these 6 amino acids. The correct biochemical way of referring to those amino acids is the 3-letter code. You don't have to write out the full name of the amino acids. At this point in time, you don't have to be able to name all 20 amino acids, nor, thankfully, draw their structures. However, if you decide to go on in biochemistry, probably at the beginning of your 3rd year, you are going to have to memorize and draw them. Yes! Okay, question number 5 asks you to compare sequence 1 and sequence 2 in order to look at what is different about them. Their difference comes up at the 4th amino acid right here. Instead of having tryptophan, we have serine. That occurs because 1 DNA nitrogenous base is different. If you take a look at the sequence, the normal strand should be A, C, C. If you look at the mutated strand, there is a G instead of a C. Because of that, when this is transcribed into mRNA, it's not the correct base. Therefore, when you look up the codon on the codon wheel, it's going to be an entirely different codon. The next one is looking at sequence 3 versus sequence 1. If you take a look at sequence 3 versus sequence 1, it's right here that you see the difference. You are supposed to go A, G, T, C, but instead it's A, T, G, T, C. In this case, you have a T that has been added where a T does not belong. If you look at how the first DNA sequence is read, that's the first 3. This is the second codon, this is the 3rd codon, etc. However, because we have an additional thymine that has been added, sorry, it's this thymine. Now instead of it being GTA, GTC, we have TGT, CAC, and everything after that point has changed. We call that a frame shift because the codon frame moves to the left or moves to the right. Finally, if we look at the 4th sequence versus the first sequence. We have a change right here. Whoop. 2, 3, 4. So we're supposed to be ACCT. We have CCT here. So there has been a deletion of the adenine. It's supposed to be right here. And because of that, the codon changes, which means the amino acid changes. Because of this, we have a left frame shift. I would love to hear your definitions for substitution, insertion, and deletion mutations.
Substitution.
All right, go for it.
One codon changes, causing one amino acid to be different from the normal amino acid sequence.
Yeah, good. Insertion. Sullivan.
I said a new strand of amino acids is introduced, added to it, or / added to an existing strand.
Okay, let's change amino acid to a nitrogenous base.
Can I say nucleotide? Or it has to be nitrogenous?
I guess it would, yes, it would have to be a nucleotide.
Okay.
So a new nitrogenous base is added to the DNA strand, which changes the codon sequence. What about deletion? What's taken away? A nitrogenous base is removed from the DNA strand, which changes the mRNA, which changes the codons. All right, now comes some logical questions, so there might be some arguing, I hope. Considering your knowledge of codons and how they code for an amino acid, would all substitution mutations lead to a change?
Not necessarily. It depends per situation.
Be specific, please.
Sometimes the amino acids will change, and other times it'll remain the same.
Why?
Because that's how the wheel works. The bottom of the wheel, like each amino acid has different combinations of nitrogenous bases.
Okay, please take out your codon wheel. If you take a look at the bottom of the codon wheel, you have a table. And that table tells you all the mRNA codons that will code for that particular amino acid. Some of them have many different codons which can result in that amino acid. So just because there is a letter change in the DNA, a substitution, which means a change here, does not necessarily mean there's going to be a change in amino acid, because there's more than one codon which codes for that particular amino acid. Would all insertion or deletion mutations lead to a change in the amino acid sequence?
Yeah.
More likely. When you take out or add a nitrogenous base, that completely shifts how the codons are read. It's more likely that you will have a brand new codon that will be read, which means a totally different amino acid, which could potentially cause more damage or a greater benefit to an organism. Substitution or insertion? What do you think, Beth?
Insertion.
Why? You're right, why?
So it's like each organism,
I want to hear the Billy logic.
each organism has a specific amount of DNA, and if you insert something that wasn't originally supposed to be there,
Kick something out.
it has to. It's not good.
Remember
Maybe lose my train of thought.
It goes outside the bounds of its own thing.
talking about Darwin's theory?
last.
And one of the key points of his theory was variation, or having differences. What is more likely to lead to a difference, a substitution or an insertion or deletion? A substitution may or may not have any change in the amino acid sequence. An insertion or deletion will have profound changes because everything from that point on could be different. If everything else in that sequence is different, that could lead to a difference in the shape or maybe a feature of the organism, which could give it the advantage to survive. So everything that we talk about this unit connects to something else that we have talked about. What do these ellipses mean?
That continues?
Yes. The reason why some of them are to be continued and some of them are not depends on whether or not you have a stop sign, right? So this amino acid sequence stops. There is no more after this. Here, this amino acid doesn't have a stop signal, so it keeps going.
So that's why the mRNA and the DNA are going on? They both have it?
Yes. The DNA will always have ellipses because it's part of such a large molecule.
Okay, and does it always have to start with an MET? Because
Yes.
mutation. Okay.
Yeah, that's the start. That's how the enzyme knows that that's the place where the DNA molecule has to unzip and start being decoded. Without that signal, how would it actually be able to determine from its environment that's what it needs to decode or do? Question 12 is an interesting question. What could potentially cause more damage or greater benefit to an organism? A deletion mutation at the beginning of the DNA or at the end of the DNA? Natasha?
At the beginning, because there's a more likely chance that it won't cause mutation.
Do you agree with her, Billy? She's right. If you have a deletion, at the beginning, that means there's the potential for the rest of the DNA strand to change entirely. Versus if you have a deletion at the end of the DNA strand, then it might only be one amino acid that is affected.
Could it be because everything has to slide over to fill the spot? It's deleted at the very beginning.
Say that again?
Okay, so in the deletion sequence,
Yes.
I don't know if it was a fluke or what, but you deleted one, right?
Mm-hmm.
And then all the other ones moved over?
Yeah. So if I took this one out,
Yeah.
then
A would go left and G would go left. Yeah.
it would, if I took T out here, then it would be ACG, TAG, TCA, CCT, AAT, which would mean the mRNA code will be different, which will mean that potentially different amino acids. Question 14. The amino acids produced from sequence 1 and sequence 2 do not have ellipses on them. However, the amino acids produced from sequence 3 and 4 do have ellipses. Why? Question 14. Why?
There are multiple new amino acids formed. There's no stop at the end of 3 and 4.
They need a stop codon. Alright, I need to scroll down to the next model. Let's just clear this ink first. Alright. There we go. In this second model, all of these mutations have had something happen to them. You need to circle the substitution that occurred by comparing the mutated DNA with the original DNA. Okay, I guess so.
Wait, which thing?
Who wants to find where the changes in mutation A?
It's,
Alright.
No,
Mm-hmm. Yeah. Hmm. There you go. Okay. So, Natasha has correctly identified that it's a substitution of this nitrogenous base. Next, mutation B. All right. So then go find where the mutation is. Mm. Mutation C. There you go. All right. Mutation D. I bet you can pick it up. I'll give you a hint just within the first little bit. Okay, this is pretty bad when the Bio 30 wannabe is doing your work for you.
Just copying other people.
All right, thank you.
Yeah, I'm smarter than you, Stefan. You took my.
All of these are a substitution of one nitrogenous base for another. Depending on where that substitution is and what codon it eventually results in and what amino acid is, it can have some interesting changes on the amino acid sequence. For this one, it is one amino acid change. Instead of being leucine right before the stop, it's phenylalanine. For the for mutation B, is there any change compared to the original DNA? Oh. Although it has a change right here in the amino acids, in the nitrogenous base letters, if you take a look at the sequence of amino acids, there's actually no change in the amino acid structure. That is because there is more than one mRNA codon for serine. This is an example of a neutral change. It hasn't resulted in any impact.
It's kind of off topic, but you know how you said if you did like a substitution or a deletion at the beginning of a chain, it could change the organism. So say if you do that on something like a cat,
I don't like cats.
how drastically would that change?
Oh.
I think.
Like would it just completely change or just a little bit?
Would it depend?
It would depend on what gene is changed.
Okay.
Is it going to turn into a dolphin? No, unlikely. It may be kind of cool.
Size or like.
It could if it affects the expression of growth hormone, for example. If the faulty gene is the recipe for something. So let's say, I don't know if you've ever used a bad recipe. It used to be in previous older generations, if you were well known for making something and somebody asked for your recipe, you would pass on the recipe, but you would, forget the secret ingredient, right? They didn't want people to be able to make this item as good, I guess. And so they would try to make this item and, it wouldn't turn out. Usually it'd be because it's missing baking soda and it doesn't rise.
No, I'm not calling them Coke.
So
My ass dog.
with this like sequence here, if we were like, would it be better to have more or less?
More or
is
less
always
what?
better, brother.
Smarties?
No, like combinations. So like, if you had more, would you be smarter or would you be dumber?
Not
Obviously, they
necessarily,
have
because
less.
Down syndrome people have an extra chromosome 21. That doesn't make them smarter.
So then
Maybe
what
we're all
if what
stupid and
if
they're
the
above
chromosomes
us.
get bigger? What would that do?
For people, it doesn't mean they're any smarter. We're the dumb ones.
Like instead of
They have an extra.
getting
Well,
more,
that's kind of what happened with Fragile X, because at Fragile X, they have a repeated section,
No,
which has elongated the chromosome, and
Because they're
that
fragile.
doesn't make them any smarter.
yeah, it definitely doesn't make them
so then what if we make them shorter?
You die faster.
Well, it
That's all
depends
it
if it's the
takes.
telomere at the end. If it's the cap at the end, it could affect longevity.
Okay, let's say the cap is fine, but the rest shrink.
It depends on what that chromosome does. I know I'm being so ambiguous with the answers, but chromosomes code for so many little things.
So then the so then the gender ones like the X and Y, what if those were bigger or smaller? Like what would happen with that?
I don't know.
Oh.
I don't know if size changes would make you more girly or less girly, for example. I don't think it's something like
What if
that.
that could that affect no, that wouldn't affect hormones.
How would it affect the release of those hormones? Because if you're increasing the size of
Oh.
the chromosome, then maybe.
Oh, I know.
So a chromosome is going to be larger or smaller because it has more nitrogenous bases in it.
Yeah.
So what is that? What is that information going to lead to? Is the question.
mental stability.
Human testing.
I can't tell you that intelligence is code CGA-T-T-G, and if we put that code into one of your chromosomes, you're going to be smarter. It doesn't. We're just starting to do work that correlates a gene sequence with a particular trait. So it's really hard to answer those questions.
I have a text that I can't remember.
The last two strands, you'll see that they cut off really early. Even though it's a substitution, and we don't think that it should have a profound impact, it does. Why? Why does a substitution for mutations C and D have such a profound impact?
Because it started earlier on, so even that little bit affected the.
Okay. By switching these two letters and having an ACT, which codes for a UGA, that's the code for stop. The ribosome is going to stop making the amino acid sequence when it gets to the stop sign. Same thing here. This is another codon, AUC is another codon to tell the ribosome to stop doing this. And so, even though we don't think that substitution should have this kind of impact, sometimes they do because that letter switch makes it a stop codon.
What does no amino acids do? What effect does that have?
Sorry?
When it has no amino acids, like the rest have like MET and GLY, what happens when there's none?
Yeah. Eventually, the cells are going to pull apart that mRNA and reuse the nucleotides to assemble a new mRNA strand. But for the rest of that, it's useless. The body's just going to recycle the mRNA for the next one.
Oh, come on.
Okay.
So, if there's no mRNA, like for the original one,
Yep.
there's no stop. Oh, there's a stop. But, okay, so what would happen after that then? With the AAAUUU? Would that
So
also be taken for spare parts then?
The stop is going to release the ribosome, and I don't think it's going to keep decoding past that. I'm going to check into that because I'm not sure if a single mRNA strand can maybe code for different proteins.
So you don't know if they'd be used for like spare parts or anything?
The mRNA, any mRNA will eventually be broken down and reused, but whether or not that UUU is going to be meaningful after that amino acid sequence is done, my understanding is when the ribosome gets to the stop codon, it physically releases that amino acid sequence. Once it releases that amino acid sequence, it can't attach another amino acid to it.
So then after it stops, like how you said that it will eventually break down and then be reused or whatever.
I'm not sure if it falls apart or if an enzyme breaks it down.
If it's not being broke down, would it eventually die?
It's not alive. It's a chemical.
Get wrecked.
Would the chemical just go away then?
Can you?
Um, the cells, from what I understand, generally don't use mRNA for energy, so it's not like it would use it as a fuel source. However, at the same time, if the cells are starving, they'll use anything.
So like, could it just like feed? Could like the chemicals? Could they like just like leave? Like say, nah, I'm out of here.
I don't think mRNA has the ability to go through the cell membrane because it's large.
What if it did?
I'm pretty sure it's stuck in the cell.
Is it possible that no amino acid is by design and it's just kind of like a backup source of energy, like how it's
Yes, because an amino acid sequence is a protein.
yeah, okay.
Whether or not the body will be able to use it as fuel depends on how hard it would be to break down that protein. Proteins don't stay as chains. They'll start folding and bending into very complex shapes. Here, I'll see if I can find them. Oh, you're going to let me finish answering this.
They're still flying. It's on your hand.
I have a bird shooting at one.
Yeah, that's what you're lucky. That's what that means.
Yeah, it is actually.
I think we're dying.
So when you get a strand of like amino acids like proline, valine, glutamine, etc., it doesn't stay as a chain of amino acids. Depending on how these different amino acids are attracted together, you'll start having really complex kinks and folds in the protein. And attractions.
Oh my gosh.
So whether or not a body could use this as a fuel source depends on how easy is it for the body to actually pull this apart and break it down. If it's going to take too much energy, then it's not going to try to pull it apart. So what they tried to do using